Hello, welcome everybody. Okay, my name is ICREA, professor of the department. So this is going to be a chance for all of us to know each other and to learn about the research that's being done in the department. Okay, I'm going to be very brief, just to welcome you. Okay, good to be here. So again, so the idea of the seminar is to foster further integration, okay, of the research in the department. So we have scheduled, okay, our talks up to the summer, and hopefully we'll continue with these after the summer, starting in October, where every professor, okay, have a chance to share, okay, the research that they do with the rest of the DTIC community, okay. So you can find details about this seminar by looking at the webpage of the department. So I'm not going to talk more. So I give the word to Miguel Angel, who's going to do the introduction of the speaker for today. Thank you.
Okay, so I would like to thank Hector for organizing this event. I think it's very interesting. And I have the pleasure to introduce Bart, who you, or most of you, all know, but it's always nice to have a formal excuse to really get to know what people do and see a bit what's hot in their topics. I brought my laptop instead of a printed version of his CV, because I thought I would break my back with all the publications. Bart is, well, he's originally from Belgium. He did electrical engineering at the University of Leuven in '98, and then went on to do a PhD, which is actually a PhD in medical sciences, also in Leuven, so from 1998, he's associate professor in Leuven. He's also associate professor in Zagreb, in Croatia, where he spent 1 year. And on top of that, he joined in 2008 as ICREA professor here in the department. One of the very interesting things about Bart's CV is the mixture of technical and clinical papers. And in fact, the translational work that he's done is really an example, I think, to the community. So he spends at least 2 days a week in hospitals. We think that is chasing nurses, actually, but he says he's working. He is, for example, associate editor of the European Heart Journal, and there are a number of appointments that are really targeted at clinical people, where he has a very prominent role. And I think this is really notorious for a person that has an engineering background. And I think I talked too much, so I would like to introduce Bart and ask him to please give the talk.
Thank you very much. Also, thank you, Hector, for organizing this thing. I think it's indeed important that we learn to know a little bit more about what people do in the department, because there's probably a lot of synergies possible. We already try to make some synergies, but I think a lot is more possible. So what I want to do today is talk a little bit about the work that we're doing in our group, the things that I'm doing, which is, as already been introduced, it's on the edge between engineering and clinical science. So I do a lot of clinical research, as Miguel Angel said, is really going to the cardiologists, talking to them, looking at patients, trying to investigate this, and trying to look from it from a more like an engineering perspective, more like looking at trying to understand physics, physiology, and trying to integrate that. So trying to combine these things. So what I want to do today is give a couple of examples of what we do and try to explain it a little bit. I tend to give a lot of information, and also you have to stop me when it's time, because I can speak forever. If there's anything you want to know afterwards, obviously you're welcome to come and discuss it. So what we do is we work a lot around the heart, well actually mainly the cardiovascular system, which is then obviously the heart and the vessels. It's mainly the big vessels that we work with. As you know, the heart is the central pump of blood for the body, pumps around the blood to the body itself, which is mainly like the head and the brain and then the rest of the organs. And the other side, because the heart has 2 sides, one side is pumping the blood around in the lungs in order to oxygenate it. So we have what we say 2 chambers, 2 what we call atria, and the heart is actually pumping the whole time, and it's actually activated or regulated by an electrical impulse which is generated within the heart, and that starts the beating of the heart so that we have synchronous beating so that the blood can be ejected at the same time towards the periphery and towards the lung. So you have this double circulation. So the heart is a pump, but it's a pump within a biological system. That means that there's actually a lot of biology going on. You cannot isolate it. You cannot see it as purely a mechanical pump, but it's really some biological thing. So it's biological tissue, and I'll come back to that later on, but it's also biological tissue within a whole metabolic, molecular environment, and these things are important to try to understand. Now also, the heart is not just like if you just take a steak or something like that and you cut a steak. A steak is relatively simple in its construction. But when you look at the heart, this is a very, very complex muscle. And it needs to be a complex muscle because what you will see that in the end, when you look at the individual cells that the heart is made of, one cell can kind of shorten or can kind of deform by about 10%, which is not a lot. So it can just shorten 10%, this cell. And with that, you have to make a pump, which actually each time it pumps can eject 60% of its volume. So by just using these cells as Lego blocks and put them together, you will never manage. You need a very complex kind of organization. And nature has developed to something which is something that looks like this, and you can see it here also, where the cells are arranged in fibers, and depending on where you are in the muscle, these fibers have a different direction. So, and the end result is that actually when the heart is pumping, what you will see is that it actually kind of screws, it kind of spirals in order to make it smaller. So it's like a sponge which is being wringed out each time, and that is thanks to the organization. Here you see the images of how these fibers are arranged, and they actually form a spiral, and then you see that the deformation becomes spiral-like. Now, one of the other things is also like it's not a very simple thing. Not only is the tissue quite complex, but when you even look at the inner side of the heart, it's not just like a piston where the blood would be pushed out, but you see that actually when you start to look inside the heart, it's very complex. You see, for example, a mesh of wires here, which actually acts first of all as a sponge to make it more efficient to pump out more blood. But what you also see is that you see all these things, these are actually electrical wires. These contain these kind of would guide this electrical impulse that starts the contraction of the heart, and to make it as kind of synchronous as possible and as fast as possible to get this contraction, we see that we have actually this whole electrical network more or less at the inside of the heart. And what you see also, like, well, the heart in itself is quite complex, and as you saw there, we also have the valves. You see here, like, these valves are also complex. It makes sure that the blood is going into one direction. You see already from this complexity that a lot can go wrong, and there's a lot of diseases that have to do with all of these things, and I will try to talk to some of them. And one of the things is also some of the research that we're doing, and that is Costa and Bruno that are doing that in our lab, is trying to see how can we describe this complex geometry. Because everybody has a heart which is more or less similar, but it's all different. And especially when you have these very, very complex geometries, you need a lot of engineering and mathematics, things that I don't understand, but these guys luckily do, in order to try to compare the individuals. Now, when we go to clinical practice, what the doctors, what the cardiologists are working with is actually ways to look at the heart, ways to try to see whether there's abnormalities in the heart. And imaging is one of the most important. And that's also what you will see is like, although I'm interested in understanding the heart, understanding the working of the heart in health and disease, one of the tools that are very, very important for that are images, because these are ways to look at the heart, to make measurements of the heart in a way that's non-invasive, so without having to do surgery, without having to destroy it. And we have several nice imaging modalities nowadays in clinical practice. The most often used is echocardiography, where you use ultrasound in order to do imaging. And you nicely see here cross-sections of the heart. You see here like the 4 chambers of the heart. You see the valves in between. You can even, with techniques like Doppler analysis, you can measure velocities and you try to visualize blood and see, for example, here that you have a lot of turbulence. So you have a valve that is leaking, actually, and this way you can try to do some diagnosis. Some other modalities is magnetic resonance imaging that gives actually the nicest kind of looking images, which is also 3D. Echocardiography is mainly just making 2D slices, although there's some 3D, but the technology is not ripe for it yet. But magnetic resonance gives you nice images, and here are examples of different kind of abnormalities in the heart. You see here a large infarct, so you see that part of the muscle of the heart is not contracting at all, and that has to be compensated by another part, so you want to try to study this. You see here an abnormally thick heart in such a way that there's almost no blood inside it anymore. This is actually the result of a genetic abnormality. So this is like a genetic disease that makes that the muscle grows abnormally. And these are, for example, the people, when you see that people are talking about young children or young athletes which suddenly drop dead, this is a typical kind of abnormality that you can have. So this is a big, big risk factor of suddenly have like the stopping of your heart, like fibrillation, and then suddenly drop dead as a young person. Here's another example where you see now, instead of this very thick heart, you see a very, very enlarged heart with actually even more complex of this kind of internal network, internal mesh. This is also a genetic disease. This is another genetic disease where mostly people at their 40s, 50s are starting to get problems. And actually, just as we can measure with Doppler techniques and ultrasound looking at blood flows, we nowadays can also, with magnetic resonance, find ways to do like really analyze the 3D flow of the blood. And you see it here coming from the atrium and actually nicely whirling around within the chamber of the heart, then in order to go out towards the aorta so that the blood can circulate within the body. Now, in reality, what cardiologists get in order to do research or to try to advance is that actually the companies that are making these imaging modalities, like every month or so almost, they come with a different way of measuring and a different way of visualizing. Of course, in order to sell their machines or to sell their software, but in the end, you get a whole bunch of information which is in a way quantitative, and you really don't know what to do with. So what ends up is that a lot of the clinical research, especially when you use imaging, is just trying out the tools that are given to the cardiologist by the companies. And sometimes it's interesting, sometimes not. Most of these don't survive very, very long. So what is, I think, much more important is try to say, like, okay, we have this technology, but we have to use the technology in a smart way and combine it with understanding of physics and physiology in order to try to further our understanding. And what I want to do for that is give you 3 examples of diseases where the heart is involved that we are currently investigating quite deeply within the lab, and which actually already also shows you that we work with a lot of the hospitals here. So this is like something on aortic dissection, so abnormalities of the big vessel of the body, and this is done in collaboration with By de Bron. Then we work actually on children, on fetal development, on how the heart is growing within the uterus while the child is developing. We do that with the Maternidad from clinic. And we actually also look at, for example, how you can use pacemakers, for example, in order to support a failing heart, and that's work that we do in clinic. And I want to go one by one over these and show you how there's the combination of clinical work, understanding, and engineering work that can help us further. The first example is what I said is diseases from the aorta, and especially aortic dissection. What you can have is that your big vessel, at some point, you get some problem, and actually all the arteries have very, very thick walls. And it can happen that there's like a slit, kind of a tear coming within this wall. And what you start to do is that blood gets in between, and it starts to make a parallel chamber, like a parallel vessel within the aorta. This is a very dangerous disease. Most people that get this, about 40%, 50% die immediately when they get it, within the first hours. If you can diagnose it and if you can go to the hospital on time, you can do something about it. But what you see is that later on these people, it's like even if you do something, like here, for example, you can do some stent implantations, but what you see is that over time, a lot of these people still have that the aorta is enlarging, 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 of course with a chance that there's rupture or another dissection. So we have to try to understand what is this process, how is this going on, how can we evaluate these patients. Now, in clinical practice, what they do is they just take images and they measure the size. That's all. So you go to some kind of imaging modality, you look at the aorta, you measure it at different places. We know some kind of values that we expect, and you see like here, in normality, it depends on the age also. You never go about 4 centimeters. So they say like, if it's 4.5 or 5 centimeters, we have to do something about it. which of course mean the size itself, depending on the person, the height, the race, and things like that, everything can change. This is a very simplistic approach, but it's an easy approach. This is the thing that we can do very easily. It doesn't take a lot of thinking. Because one of the things that if you start thinking about, okay, how is this process of this abnormality, both the formation of this abnormality, the formation of these tears, but especially the further dilatation of this vessel over time, how is this taking place? Well, one of the things is obviously that you have to start thinking about mechanics, because there's a high pressure, high blood pressure inside, which is cyclically deforming this vessel. So looking at the wall stresses that are induced is obviously important for when it's going to tear and when it's going to rupture or when it's going to grow. Now, the other thing which is also important is actually that there's flow, continuous flow, well, pulsatile flow in between. And we know that when we have flow, which is viscous, because it's like kind of biological flow, what you will have is that at the sides, at the boundary of the vessel, you get shear stresses. So you really have like the fluid kind of takes away a little bit the vessel. So there is like small stresses at the inside, what we call the endothelial, so the inner side of the vessel. And what you have is that actually the vessel wall has a layer of single cells. So there's one layer which forms the inner layer of every vessel, and that's one cell thick. And that's what we call endothelial cells. And these endothelial cells, what they like is that the flow goes in one direction, and they're nicely elongated, and they're happy with it. At the moment that flow is changing, that it becomes turbulent, that it becomes too high or too low, what happens is that these cells don't feel well anymore, and they start to deform. And they start to, instead of elongated, they start to become rounded. And the most important is that actually these cells are very active biologically, because they release a lot of molecules, a lot of growth factors, a lot of things. That means that if they're not in a happy environment, what happens is that they start to release growth factors so that the vessel starts to dilate. So that means that the biology determines how this is going to go, but the flow, the local flow profile, and here you can see that this flow profile can actually be quite complex with a lot of turbulences, with a lot of vortices. This flow profile, together of course with the pressures and the wall stresses, this is what determines in the end the mechanics. So that's the thing that we have to look at. Now, of course, the clinicians realize that, and they say like, okay, let's do some research and try to see how flow can help us in understanding these patients. And what we can see, as I said before, we can measure flows actually with magnetic resonance, for example. So what we see is we can have some kind of flow patterns, and then you get for one patient, you see like, okay, how is the flow going in the vessel? How is it going when we have this dissection, for example? How is it going in the real vessel? And how is it going in this additional, what we call like the false vessel or the false lumen? You can measure this in a lot of patients, and what you then get is, okay, the clinicians then talk to us and say like, okay, can we do some analysis? Then you write a couple of MATLAB scripts where you can try to see, okay, can we get some kind of properties of these curves that would correlate with what we find in the patients? And then you can make this kind of nice statistics. You plot all the patients when you have a lot, you do some thresholds, and then you find that indeed there might be groups looking at these patterns which behave differently. Of course, this is just the analysis of the data. This is like fancy statistics, but most of medical research is fancy statistics. It's not trying to see, okay, how can we go further? How can we go and interpret this better? So then, of course, we get in the engineers, and this was like the PhD project of Paula, which is still working in our group. And what we then say is like, okay, let's try to understand what's going on. And what we do then is we make a setup in the lab. We make some kind of phantoms here based on latex, and we say like, okay, let's now measure these, let's measure pressure, let's measure flows, do that in the lab. Sometimes they explode and the whole lab is wet, of course, but okay, at least we can try to understand it, and we have measurements of velocities, of pressures, and we try to see, okay, can we now look at the dynamics, the mechanics of the system, and try to see whether we can understand some more. And nowadays, what we also can do is, of course, do computational simulations, and these are quite important. Here you see this like fancy computational fluid dynamics. But one of the things that we will see also is like actually, because this is a rigid system, and the biological system, especially the aorta, is actually very flexible. And what we quickly realized is that actually this is not good enough. This is not representing what we see. So we have to try to get further, and we have to try to understand a little bit more. And actually, sometimes, like, what you need to do is then go back to the data and say, like, okay, which are the things that we see in reality? Can we try to see some patterns that we can then try to see whether we can understand them better? And when we look at our patients and we look at these flow curves, what you actually start to realize is that there are indeed some patterns. And what you see, for example, is if we here look, this is like the ejection phase of the heart, so the beginning of the cycle. What we see is that this can be either like biphasic or it can be monophasic. Or when we go to when the heart would be filling, so what we call in like the diastole period, what you see is it can be either positive or it can be very negative, which means that the flow is going either in this direction or it's actually going retrograde, so it goes, instead of from the heart to the body, it would go from the body towards the heart. And these are the things that you see. So we tried to understand this, but we realized quickly that with these fancy computational fluid dynamics, we cannot do it. Because very likely, especially for these retrograde and for these changes, elasticity becomes quite important. So what we do then is also, sometimes instead of doing fancy research, it's sometimes important to go back to simple research, but that you know where it is and that you say, like, okay, there we can really understand and get conclusions. And one of these things is that you can actually make also simplified models of hemodynamics and hydraulics. And this is a thing that we do here. So what you see is we have this abnormality, we have the vessel, the aorta, we have these kind of tears with the second kind of chamber. And what you do is you make actually an electrical equivalent model for the hydraulics, where you use resistors, you use inductances for looking at viscous flow, and you use capacitors, and that's the most important in this case, in order to kind of cover the elasticity in a way of the vessels. And when you start doing this, you can start to simulate, okay, what's happening depending on the geometry and the properties of this tissue. What you see is that looking at the elasticity, what we see is that, oh, flow is not changing that much in itself if the elasticity changes. But keep in mind, it is still elastic, it's not very, very rigid. But what is important is actually when you, depending on where you measure, you see actually something totally different. When I showed you these images or these curves that the cardiologists are measuring, they said, but let's measure at this point, without thinking that maybe it matters where you measure. So again, luckily they do it always at the same point or more or less standardized, so you still can get some conclusions out of it. But if they by coincidence would have done it a little bit higher, a little bit lower, you would have different results. So these are things that we say like, okay, maybe we should think about it a little bit better and do other ways of doing. Another thing is also depending on how big these tears are, where these tears are, we see that we have changes in these curves. And so by making these combinations, we see that indeed we can simulate this either biphasic kind of response or this positive or negative flows. And so what we now can start to do is try to interpret the measurements and saying like from the measurements try to infer information on the tissue, on the geometry. So these are some of the things that we have to do then further. One of the things is then, of course, as we said, what you can do is you can nowadays also do simulations with elastic walls. However, these are very computationally expensive and more difficult, but we will try it at some point. And what you also see is that you can do these 3D measurements of flow and then try to, for example, couple your flows and your simulation. So these are things that we are thinking about in order to do in this direction. A second application that I want to go into a little bit more detail is what I told you about, is that I work also like 1 day a week in the maternity hospital, and we have several projects with them. And what they are doing is they're trying to investigate and see whether things that happen during pregnancy, whether that can influence what the fetus or what the person will be later on in life. And one of the things, well, actually a lot of research in this area started also with this finding, is that at some point one epidemiologist in the UK realized that when he started to look at cardiovascular disease in a population of 60, 65-year-old people, and he looked back, I don't know how it came that he looked back to it, but he looked back at infant mortality when these people were born, what you see is that there's a very, very good geographical overlap. So they started to think, it's like, maybe something that happened when you were born determines what problems you might have when you're like older. So, and then people started to do research, and then when you start to look in cardiovascular diseases, we all know that when cardiovascular diseases, everybody says, like, in order to prevent cardiovascular diseases, you have to live healthy, don't smoke, don't eat too fatty, and things like that. You might be unlucky that you are in a family that has a lot of cardiovascular diseases, but actually what we start to realize is that actually during the pregnancies, things might happen that determine whether you will get problems 50, 60 years later. And one of the reasons is also when you start looking at the problems around pregnancy, when does a fetus have problems? When is a fetus maybe going to be stillborn and cannot survive the first days? Well, there's a couple of things which are important, but one thing is what we call intrauterine growth restriction or fetal growth restriction, which actually means that the placenta of the mother, instead of like this with a lot of vasculature, looks like this and is starting to be insufficient. So that means that especially at the last weeks of pregnancy, when the fetus wants to grow a lot, the placenta is just not big enough, not good enough in order to provide the oxygen and provide the nutrition. And so the fetus is restricted in its growth. And it will have not enough oxygen, not enough nutrition. And what we see then is that actually there starts to be adaptations in the fetus in order to try to cope with this. And some of the things that you see is like when you look in clinical practice, actually, the way that they currently kind of determine this is, in general, they do it when you have a baby which is within the lowest 10% in birth weight. So when you have a very, very small baby, then the chance is very big that the baby was growth restricted. So in most clinical practice, they say without the lowest 10%, they potentially have problems. But of course, this is in a way simplified. You can be small indeed because you didn't have enough nutrition in order to grow, but you can also be small because your parents were small. So in that sense, it's like, okay, which is really going on? And then one of the things that you can start looking at is trying to see, okay, what is happening when we have this growth restriction? And one of the things that you see what's happening is that actually what you see is, well, the placenta is insufficient, which means that probably the resistance of the placenta is going to change. And of course, if the heart has to pump through a placenta with a higher resistance, you need to do that at higher pressures. So that means that your blood pressure, or the blood pressure of the fetus, is going to rise. The other way around, if you have less oxygen in your blood, a way to provide oxygen to your organs is just by pumping around a lot more blood. That's, for example, when you do exercise, you need more oxygen to your muscles, so you start to increase the pump volume. And that's what you see. So because of this abnormality, you see that both the pressure and the volume is changing. So you get an overload of the heart. And one of the things that you also see is like when we look at the fetal circulation, in the fetus, the lungs are not working. Of course, because the fetus isn't in an air environment. So at that moment, the lungs are actually kind of shut out of the circulation. And they're partially shut out by the fact that you have actually a connection, which is called the ductus, between the lung circulation and the body circulation. While in a normal person, they are totally split. So you have one side through the lungs, one side through the body. In the fetus, because the lungs don't work, you have to do something, and there's actually a shunt which closes when you're born. When you take this shunt, in a normal fetus, what actually happens is that the blood that should normally go through the lungs goes now through the shunt, and then goes further towards the aorta and towards the body. That's the normal way where that blood that should go to the lungs goes. However, if this placental resistance is much, much higher, and the fetus is having problems with oxygen, one of the first reactions is that the fetus tries to protect the brain, because that's the most important organ, of course. So what happens is that actually the vessels in the brain start to enlarge so that more blood can go to the brain. And what you see now is that actually instead of the blood that should go through the lungs that would go in this direction, it actually reverses and it goes towards the brain. So you see a flow reversal because of this placental abnormalities and because of the fact that the fetus tries to protect the brain. And you can actually measure that with some Doppler techniques. And you see then here that you see like normally flow would be in one direction. In this abnormality, you see that it's in the other direction. So now you already have a tool which is a little bit more sensitive than just saying, okay, the lowest 10% in birth weight. No, the lowest 10% or maybe the lowest 5% plus evidence that you have hemodynamic changes that are induced by these abnormalities. Now, of course, then it comes in, it's like, okay, what are the consequences of this? And at some point, the obstetricians, like the first time that I started to collaborate with them, they came and said like, yeah, we did here a lot of measurements on these fetuses, and we see that the heart seems to work differently, but we don't understand it. I said like, okay, let's have a look at it. But then I said like, again, the engineering reflex is like, okay, let's try to understand what's going on. As I said before, you have a higher pressure, you have a higher volume within the heart. What could be the changes then? And one of the things is that, of course, then you start to think again about mechanics, and you know Laplace law, which says that if we have an object and we want to know the local wall stresses there, well, the wall stress is related, of course, obviously to the local pressure, but also to the radius of curvature, namely to the shape of the object. If we have a very small round object with the same pressure, we have low wall stresses. If we have a big object with flat walls, we have much, much higher wall stresses. And so you actually see that when you look at the shape of the heart, a normal heart is very elliptical, because an elliptical heart is very efficient to pump blood. However, if, for example, your pressure rises, what you get is, of course, you can lower the wall stress by becoming more spherical. So you see actually that when you, for example, look at a normal heart, this would be a normal geometry, and when you would suddenly have a blood pressure increase, for example, when you go to the toilet, you kind of push, you increase your blood pressure, how your heart actually reacts to try to minimize this overload of the wall stress is by slightly changing its geometry and becoming a little bit more spherical. So if we have fetal overload, if we have fetal pressure overload, maybe something is already happening to the shape. And indeed, when we started to look at these fetuses, what we saw is that these growth-restricted fetuses start to become much, much more spherical compared to normal. And what we actually now also found out is that this is being done in fetal life, but your heart is still forming in fetal life. So that means that you're born with a heart which is more spherical, and actually you always keep a more spherical heart during your whole life when you're born with it. And of course, when you have a more spherical heart, when you go to the toilet, you cannot make it more spherical even. So that means that relatively, you always have slightly higher wall stresses within a heart, which is when you're born like this. So you're born with a less efficient heart. Although, of course, once you're born, there's nothing wrong with your circulation anymore. But by the fact that your heart is formed during fetal life, you see that there's a difference. And you actually also see that even your vessels, your vessel walls become thicker because of the fact that you had during development a higher pressure. And we all know that thicker vessels means you have a higher risk factor for high blood pressure, and high blood pressure is also a cardiovascular risk factor. So in fetal life, it's already determined that later on you're going to be potentially more in trouble because you just have a less efficient cardiovascular system. Now, if you want to understand more than, okay, if we say this shape change, okay, we see this shape change, but there's more going on. Well, as I said before, the heart is quite complex because when you look at the tissue in itself, the macro shape is important, but it's built of all single cells, as I said before, which are the contractile material. Within these cells, you have organelles, and there you have the molecules which also can make that your cells can contract. So when you want to understand remodeling, you can be sure that there's highly likely remodeling at all these levels. And so when you try to see is like, what is going on in these diseases, you cannot just say like, okay, let's do measurements and see statistically what's their difference. No, we have to try to understand, and we have to go actually to all of the levels, to all of the things that are going on, going from some of the fiber orientation, for example, looking at the myocytes, what they look like, so what the cells look like. There's actually probably differences in electrical activation. So that means that you have to do really multi-scale research if you want to get an overview, if you want to look at what's going on. And that's exactly what we are doing in the maternity hospital, where we look at, well, first of all, what you do in biology is also to mimic diseases in a way that you have more control over it. What you try to do is you make animal models of this disease, and that's also what we have. We have, for example, a rabbit model or a rat model where we can do that. And obviously, you can also look as much as possible to fetuses, to human fetuses, but of course, there you're limited in what you can investigate. And what we then try to do is looking at the organ level, looking at the cell level, and even looking at the subcellular level to see what's going on. And some of the things that we have found, which is a combination of doing imaging, in this case microscopy, but also using some kind of tools being developed by the engineers of our lab in order to do the analysis, what you see is that actually not only is the shape of these hearts different, but the individual cells are different. Within the cells you have differences of the organelles, and even at the contractile proteins, you see that you actually have shorter proteins when you're born like this. So you see that indeed at each level, by doing imaging and image analysis, you can show that there are differences. And then, this is just big fun. This is something that we are trying to do. You probably heard about the synchrotron already, or at least about the Barcelona synchrotron, which is this one, which is in Cerdanyola. And when I read about it, that it was there, I said like, this seems like fun. I want to do something with it. And actually, you can do quite a lot with it. And I found out that for the heart, you can actually do really a lot with it. Just very briefly, the way a synchrotron works is what you do is you take a ring, you start to accelerate electrons up to light speed. What you do is, in order to keep them in a circular orbit, what you do, electrons are of course charged in a magnet which is banded. The electrons would follow the magnet. So electrons follow a magnet. However, when you take an electron and you force it to bend, what happens is that it loses some of their energy in the form of photons, in the form of light. And of course, photons are not charged, they're not banded, so the photons, they fly straight out. And what you can do now is each time when you have these photons, you get like a very intense bundle of light, and it's actually very synchronized light also, and you can try to do something with it. And part of this light is actually X-rays, and a lot of the imaging that we do in medicine is X-ray imaging, especially computer tomography. With X-rays we can make kind of nice views through tissues. And one of the things that you do is like, this is a typical example, what you can do is non-invasively at high resolution you can look at it. And this is like paleontology, so these are like two dinosaurs which at some point fell asleep and then were embedded in rock. And so this way, non-destructively, you can really look at that. Of course, besides dinosaurs, what I do is we put hearts in there and we try to image hearts. And this is already the image that I showed you about these fibers. This is actually done in the synchrotron, where at a resolution of the order of magnitude of like 5 micrometers, you can image like whole hearts, in this case of a rat. And you see all the details of the structure. And once you have that, you can try to quantify it, and that's what we then also did. We tried to look at the difference between abnormal growth and the normal growth. And here you see that within these images, for example, you can extract the vessels. So that's work from Chong, another postdoc from our lab. What you can see now is that when you look at these abnormal hearts, you see it here already, is that actually the vessels that bring the blood to the heart itself, they're much, much more dilated. Obviously, because there's not enough oxygen, so you need to even to the heart provide more oxygen, and you do that partially by getting bigger vessels. But that means, of course, that the amount of vessel here is much larger than the amount of contractile material, so that again might compromise your cardiac function. So these are the things that we try to quantify. And these images are of such quality that we can even try to extract, and we are doing that. This is actually a PhD student that I collaborate with in Zagreb University, and she is extracting these fiber orientations, and then we can really quantify in very, very much detail how this local fiber orientation of the heart is there. Now, as I said before, this is a disease that involves the whole cardiovascular system, so we have these reversals in the vascular parts. And what we try to do there is, of course, in order to understand that, we try to again use some computational modeling to do it. This is some work of Patricia in our lab, that's her PhD thesis, where what we do is we take now the whole fetal circulation with the brain, for example, but also with the placenta, with all the different vessels, and we then make a more kind of, in brackets, complicated model. It looks complicated, but in the end it's like all the same building blocks. But what needs to be done is, of course, you need to calibrate this, and that's a little bit more difficult. But when you do it properly, what you can see is that you can really use this kind of tools in order to simulate the measurements that we do. So these are like flow measurements in real fetuses done in the clinic, and you see that we can nicely kind of get these flow patterns, that we can simulate these also. And we have actually quite a good agreement. And once you have these models, then the system crashes. Sorry for that. So once we have these models, what you of course can start to do is try to study the model in order to try to get more information from it. And this kind of things, what you can do is you do parametric studies where you see like, okay, at which point do we start to get flow reversals? What's the influence of the brain resistance, the placental resistance? It's like what has to change in order to get the things that we see in clinical practice. But even more nicely is like when we have good models, what we can try to do is use models also to calculate or estimate parameters that we cannot measure in real clinical practice. For example, as I said before, it's like this growth restriction is mainly because that there's an abnormal placenta. And the resistance of the placenta is changing, but depending on what is exactly wrong with the placenta, you will see that the compliance is also changing. But resistance and compliance we cannot measure. But we can try to simulate or use our simulator and try to use an optimization algorithm to fit data that we can measure from the fetus to the model. And then based on the model, you see here some of the fittings, so that goes quite well. But then we can use the model to estimate indeed these resistances, for example, and the compliances. And what you then see is that indeed, of course, we know there's a change, but you can quantify the change. But what is actually more interesting is if we calculate the resistance and the compliance of the placenta, we cannot only say that there is a growth restriction, because we have already things to do that, namely looking at flow reversal and the estimated weight. But what we actually can also predict is whether there's adverse perinatal outcome. That means, is this baby potentially going to be in trouble during birth? If you know that there's a much higher chance for trouble during birth, of course you can try to prepare for that. And you can do, for example, a cesarean section, or at least know that there might be trouble. So this way we can try to predict things that are very, very difficult otherwise. As last example, I want to talk a little bit about this is like, what do we do next here? Well, next is, first of all, we're starting to simulate cells, see the individual cells, the mechanics of the cells, that's what we do. Another thing that we try to do is playing a bit more with the synchrotrons, and what I now want to do is put a beating heart and see how fibers are changing during the heartbeat. Nobody has done that before, so that's another nice challenge. And then we need to model the placenta and the mechanics of the heart. So the last example that I was going to talk about is this electrical abnormalities in order to try to treat heart failure. And what you see there is that currently there is like, so you have a lot of people, like especially elderly people, that have heart failure. The heart is really failing. And that means that most of it, your life expectancy with heart failure is maybe 1 or 2 years. So that's one of the major causes of that in our world. Now, of course, you try to see what you can do for it. There's a lot of drugs for it. But what people found out is actually that in several of these heart failure persons, what you have is like when you look at the cardiac activation, like the way the electricity is being propagated in order to do this kind of synchronous contraction of the heart, there's something going on there. And some of these kind of networks of electrical cables are actually cut. So that you have a cut here and you don't go anymore through all these cables that I showed you. The result is, of course, that the activation of the heart is not simultaneous anymore. It's not happening at the same time anymore. And you start to get what is called dysynchrony. I'll show you a little bit later what it means. But it means that this heart is not efficient anymore and will start to fail. What I found out is that actually when you take pacemakers and you start to give electrical impulses at some places, and that means that you can much faster activate the heart, you can actually revert that. And these pacemakers, they cost about 30,000 euro. And what they have found, that in about 70% of the patients, when you select them as saying like, okay, there is something wrong with the activation, there is, well, the patient is having symptoms. It's mainly the patient cannot walk anymore on the stairs or something like that. And if you look at the heart, it's like instead of nicely going like this, it actually goes a little bit like that. If you see that in a patient, there is a chance that this patient will respond. So these are really the criteria that we have now. We don't have anything better. We haven't found anything better. And so we put a 30,000 euro pacemaker in this patient, and what we see is that 70% of these patients, they really, really get better, but 30% actually gets worse. And we don't know why. At least not using these criteria. And there has been a lot of research, again, by doing measurements and doing statistics and trying to say, like, okay, this parameter might work or this parameter might work. But you can already imagine also that there's not a lot of kind of stimulation from the industry in order to make this a bit more efficient, because of course these 30% of pacemakers, still a lot of money for the industry in order to do that. And one of the things that you see is that medical doctors, they try to make their life as easy as possible, in the sense that they say, like, okay, I take an image and I calculate one parameter. Is this parameter above or below a threshold? I'm going to do something. Because of course that's easy to interpret. But especially something like a failing heart, when you see a heart which does this instead of that, I mean, there's of course a million reasons for it, given the complexity of the heart. So that means that just by trying to find one parameter which would fit it all, for sure you're going to get in trouble. And that's actually what we see also. And that's one of the reasons why we cannot do anything better. So again, what you need to do is, of course, go back to the mechanics, try to understand what's going on. And what you do there is like, okay, we start to say, is like, if there's an electrical abnormality, what's happening now? Well, it means actually that we don't have this kind of synchronous contraction anymore from all the walls, but what we see is like there would be one part of the wall which is activated first and which would start to contract and actually might start to stretch surrounding tissue before that tissue is being activated. And that's actually what you see is if you look at this heart, like a normal heart would really go like this, everything synchronous. But what you see here, and especially when you slow it down, you see here like this very fast, like inward, outward motion. You see it here up, inward, and then everything is like turning a little bit. This is definitely mechanically abnormal. So trying to understand this, trying to see whether we can use that, that's one of the things that we also try to do. And what you actually see is that when you start to look specifically for this, and here is a patient before and after this pacing, you see actually that this very abnormal motion, you really get rid of it when you put these pacemakers on. So you can really correct something which is mechanically abnormal. And that's one of the things that we try to understand. And one of the things that you also see, and this is actually a little bit funny, is like if you start to look at how this electrical activation is taking place, it's like what you expect is like, okay, you have somewhere a pulse generator, and then that goes, you have conduction of the electricity, and this electricity conducts either fast if we have these very fast wires, or when we don't have the fast wires, it will go cell by cell, and it will go much slower. But we expect this to be in a way homogeneous, because we know the propagation of electricity in a medium should be a kind of a diffusion process. However, when we start measuring things, what you actually see is that in these patients, so they get an electrical impulse, part of it is activated, and suddenly everything stops. And for quite a long while, there's no conduction anymore, and then suddenly it starts to conduct again. So you start to say like, okay, there's something going on. If you ask cardiologists, what they say is like, no, you have electrical activation of the heart, it activates everything, and then it starts to contract. These are separate things. Activation is fast, contraction is slow, so we just study them separately, and you have separate specialists. You have electrophysiologists, and then you have heart failure people. They don't talk to each other mostly. But when you start to do these measurements, you see, well, there's something strange there. And then, of course, we again come in as engineers, and we start to say, like, maybe there's something else going on. And what you now actually see is that if electricity is slow enough, what you can get is that mechanics can be faster, and you can stretch tissue before it's being activated electrically. And what people have been doing when they do cell work is actually a total different environment, but when you look in the literature you can find it, is if you take a cardiac cell and you stretch it, you actually change the ion channels, so you actually change its conductivity. And so by stretching tissue, you can actually make it non-conducting instead of conducting. So you change the conductance speed by doing that. And this is what we simulated here, so you can indeed have that if your electricity goes too slow for some abnormality, the mechanics can catch up, stretch tissue, and change it. And then, of course, when you go to clinical practice, it's not anymore we do one parameter, or we say, are you feeling ill? And looking at the ECG, is that abnormal? No, it becomes, of course, a lot more complex. And we have to try to find ways to specifically look at which are the mechanical consequences that you go, and you start to make this kind of decision trees. And Additionally, it's like life is not simple at all. I'm not going to go in detail here, but again, this black and white doesn't exist because there are a lot of things which are important. It's like, for example, when you give electrical therapy, there has to be obviously something electrically wrong. But again, it might be that you already have a very bad heart with a lot of infarct tissue and so on, which is actually scar tissue. Of course, that's not going to contract anymore, so that will play a role. And additionally, putting this pacemaker is actually also a little bit tricky because putting these wires and so on, sometimes it just doesn't work. Sometimes you don't manage. So there's a lot of factors which actually will determine whether you can respond to this therapy, yes or no. So this 30%, which gets an expensive pacemaker, is actually a very, very complicated population. And we have to do that in a much more kind of elaborate way, thinking about the physics. And does this work? Actually, in hospital clinic, together with Marta Sitges, we have indeed proven that it works. And so what's next there? Again, trying to simulate more. And we actually currently in a European project that is trying to investigate this in a little bit more detail. So, in conclusion, what I try to tell you is like, clinical practice, while the doctors want to make it simple, it's not simple. But the way to try to understand it, first of all, to understand it, understanding is one thing, but also then try to translate it into diagnostics or therapy, is that you actually have to integrate information. Not only do you do clinical measurements, but you need to use physics and mathematics, you need to use simulations, you need to use engineering tools, even make phantoms with water and things like that. And you need to also, quite importantly, try to understand basic physiology. When you do cardiac work, you need to know how a cell is working. Maybe the cardiologist doesn't know it, but when you try to help, you have to try to put all these people in context and in contact. And this is actually trying to integrate all this information is what we try to do in our research group. And as we said before, research is never done alone, and so I want to thank everybody of our group and all collaborators. Thank you very much. Too much information.
I ask a question. So part of what you do, you generate a lot of data for physicians. But as you say, people sort of cognitively are poor, so you cannot handle a lot of data. So at the end, they want to apply policy, say, give me just one parameter, one variable, if it's above the threshold. So how much of the data is interpreted by machines automatically? How much of the data is interpreted by people? And how this division should work?
Yeah. Currently, everything is interpreted by people. There's very few like machine learning, artificial intelligence approaches that are currently used in clinical practice. There's two reasons for it. First of all, it's complex. But secondly, I think also the artificial intelligence people in the past have done it wrongly. Because what they mostly do is they say to the doctor, I come here, I give you a system that makes your decision. I mean, if you say to a doctor that somebody else is going to make a decision, then they start to get worried. It's like they're going to lose their money, of course. They don't like it. So, because, I mean, even the parameters that they use, even if they're stupid, at least they're a little bit used to it. They don't know exactly what it does, but they have a feeling for it. If you give just a black box, they're never going to accept it. So what we start to do now is actually, and this is, we are collaborating, for example, with Anders for this also, is trying to say like, what you start to see is you start to do a little bit of data mining, but in this case, not in order to generate a black box which would give you the answer, but to try to say it's like, maybe you should look at the data in this way, or maybe you should look at it in that way. And so I think these approaches will work at some point. And we're trying in the lab kind of several approaches. For example, so then with Anders, Gemma Piella is also doing, I think when she gives a talk, we will see a little bit more about that. So I think it will slowly start. What you actually also start to see now, for example, is there's I know of the hospital that I used to work in Leuven. What they're trying to do is at some point they were saying like, okay, we have people that come in intensive care and they get kidney problems. They're expensive. We need to see what's the chance that they get kidney problems. And then there was one guy that did a PhD in trying to see what this was and trying to say, oh, this can happen, that can happen. And he was reasonably good at interpreting the data. And then the guy in the hospital said, which is now in the informatics department, said like, what if we give all our data to Google and see whether they can do better? And so they gave like data from like 50,000 patients or something like that to Google. Google came up with a black box and was actually much better than what the PhD student did. So there is a lot in current technology of like deep learning and data mining and things like that. But we need to find a way to translate this into something that can be understood and try to give it as a guide or an aid rather than as a decision system.
But even if it's not understood, its performance can be shown to be much better for a black box system, okay, that you don't understand. The question is, would you like to be sort of diagnosed by a guy that can explain, I mean, his reasoner, or by a black box? Okay,
in the end,
it's a much higher performance.
Yeah, in the end, no doctor will like the black box, because also, well, what you see is, yeah, but the thing is, what you still want is that there's a little bit of the knowledge, because also with these black boxes, you don't want to be in the 10% or 30% misclassified. You want to say, like, maybe there's something else that this guy can use in order to do it properly. So I think people are finding out what you can do with it at this moment, but it's definitely interesting research. Yeah, so that's indeed what you need to do. You need to try to extract the thinking of the black box in a way to see how it fits with physiology and things like that. So that's the way forward. But that needs a little bit of change also in these algorithms, because currently these algorithms don't care. What you need to do is try to combine data mining with a little bit of knowledge and try to see how you do that. Ralph? Yes, yes. Well, what I was talking about was desynchrony of contraction. Contraction needs to be as synchronous as possible. That's always mechanically the most efficient. What has to be desynchronous in a way is your heart rate. Because what you see is when you, for example, look at heart rate variability, when you see that patients that have during the day constantly the same heart rate being very, very synchronous, they're very, very bad and they're very, very sick. Because when you change, when you stand up, when you walk, when you sit, you need to be able to adapt very, very quickly to changes in volume that you need. So you need an unstable or chaotic system in order to be able to function properly. So in that sense, is for heart rate, you need a desynchronous system, or a balance at least between synchrony and desynchrony. For the mechanics, synchrony is the best. So Michelangelo
also elsewhere at UPS in the
yeah.
Environmental Sciences Department.
Yeah. With Michelangelo we work very, very close. Our two groups are actually one unit in a way. So there is a lot of synergies. As I said before, we started to do some machine learning with Anders. We're working with Leo a little bit in trying to get this kind of this data mining, but then at an even higher level where you look not only just as measurements of images of the heart, but where you take all the data from a patient and you try to do some kind of knowledge extraction and things like that. So these are the things that we currently are doing. For the moment, with the sex department, we don't have so much synergies because they do mainly basic research. And I'm a little bit more interested in like the basic/clinical research. So for the moment, Hospital del Mar is still starting as an academic hospital and the cardiology department there is not strong yet. So that's why currently I'm mainly working with clinic and by de Bron at this moment. So that's where most of the synergies are.
Okay, just complementary on Angel's question. Are there other groups, other research lines that you would like to hear more about that you see a potential, okay, that currently you don't have any collaboration, but you'd like to hear more than you think?
Well, definitely. One of the things is like, for example, sometimes data visualization or data mining, that kind of complex ways of working with it, that is something that is quite important. Visualization also, these are things, so there are things that could be important. And the other hand, there's also, at the moment we don't do it a lot, but for example, ECGs is like signal processing and signal analysis, and there with the music group, there's probably also some kind of synergies possible. Because at some point we were actually already vaguely kind of talking at some point with people. What we do is also try to do spectroscopy in order to look at the metabolism of the heart, but spectroscopy from magnetic resonance and music analysis, there is some overlap there also. So I think by learning more about what other people do, you can say, maybe this I can use or that I can use. Or at least it's good fun to know what people are doing. So.
Okay, thanks a lot, Bart. So thank you, guys. Okay, just one point of information for faculty, don't get lost. Okay, so for those of you who are only, sorry, okay, we cannot invite everybody. Okay, just upon the faculty, take your track, ICREA and whatever. So there is now a following joint lunch in room 52-421. There is double ration for the speaker. Okay, thank you.